Hi, I'm Amelia Marani, and today's lesson is ion channels in neurons. On the next slide, we'll look at an outline of how I'm going to proceed today. We'll begin by looking at the neuron and the parts of the neuron, that cell type where our channels are located. Then we'll look at different types of channels, and those will include voltage-gated channels. We'll look at where those are and how they're activated. Then we'll turn to ligand-gated channels. We'll look at how those are activated and where those are on a neuron. And then we'll look at leak channels, and we'll also look at how those are activated and where they are on the neuron. Armed with an understanding of the three different types of channels, we'll turn to the expression of those channels. We'll look at the protein structure of the different channels, and we'll look at how that structure enables the functioning of those channels. All right, let's begin. So this is a drawing of a neuron, if you will. At the input side are dendrites, and dendrites are integral with the cell body. And inside the cell body, we see there's a nucleus. Then there's a long axon connected to the cell body. The axon has myelin sheaths along its length, intermittently, not continuously. And in between each of these myelin sheaths are nodes of Ranvier. And there's a Schwann cell located in each of the myelin segments, if you will. The neuron ends, this particular neuron type, they're not all exactly the same, with axon terminal, and we'll talk more about that as we proceed. Let's begin looking at voltage-gated channels. Turning to the left figure on the left, we have a lipid bilayer where these beads here represent lipophilic heads on the cytosol side or intercellular fluid, and these beads represent the lipophilic heads on the extracellular side, and extending there from each towards the center of the bilayer are the hydrophobic tails. Sodium isn't a sodium atom that's generating the sodium current through voltage-gated channels. It's a sodium ion, which means it's positively charged and it's lost an electron. In turn, even though its size is not prohibited for it to be able to cross the cell membrane, the polarity prevents it from doing so. That's why it needs a channel. But where a channel present, sodium could move down its concentration gradient from the extracellular space to the intercellular space, where extracellular sodium concentration is near 14-fold greater than sodium concentration on the cytosol side. However, the channel is not normally open and has what's called an activation gate that is voltage sensitive. So the membrane has a potential, or a difference in potential, as between the cytosol side or the intercellular fluid surface and the extracellular side. And here we've drawn some negative charges, just representing the fact that this potential on this side of the membrane is more negative than the potential on this side of the membrane. And cells have a resting membrane potential. That is to say, when there's no electrical activity going on, cells are at near minus 80 millivolts. So the intracellular side is more negative than the extracellular side. And when the membrane potential is at minus 80 millivolts, the activation gate for sodium is in the closed position. In turn, sodium is enabled to get down into the cell. Now, as membrane potential rises about 20 millivolts, this gate will swing open, and now sodium, remember it's positively charged, can move down its concentration gradient into the interior of the cell. And this has an effect not only to get sodium from one place to the other, but it's an electrical charge that moves in, and that raises the intracellular membrane potential. It gets closer to that of the extracellular space, and as it gets closer and closer, another channel can be activated, and that's the potassium channel. Now, potassium has a greater concentration on the intracellular side relative to the extracellular side. So if we drew potassium as little purple circles on the intracellular side, we would have a lot of potassium on the intracellular side, and we would have just a few on the extracellular side. And until the membrane potential reaches a significantly less negative or positive potential, the activation gate for potassium is closed. But once it gets to about 0 millivolts, then potassium will go down its concentration gradient. And just as sodium going in depolarized the cell, so if this were resting membrane potential, the potential would be rising with the entrance of sodium. Then with the exit of potassium in the opposite direction, that's going to serve to repolarize the cell and bring it back to what's near resting membrane potential, which was about 80 millivolts negative. All right, let's move on and see where these voltage gated channels are located. So both sodium and potassium ions and their channels, not the ions, but the channels for those ions are located at the nodes of Ranvier. They're co-located. So an action potential will take place here that will actually depolarize this area enough to where another action potential will be generated here. Sodium channels and potassium channels, sodium channels first, and then potassium channels open, depolarize, potassium channels open, repolarize, and the action potential will quickly propagate down the length of the axon with potentials bopping along at the nodes of Ranvier. How are these voltage gated channels activated? What makes them move? Well, remember the sodium ions themselves, the green dots, were charged, and then we placed charges above and below the membrane to indicate there was a potential, and in fact there's charges on each side of the membrane. Well, there's also charges in the gate themselves, charges in the gate, and any two charges are going to induce a force on each other. So this is capital F for a force, and if the charges are the same polarity, so two positive charges, for example, two sodium ions, those are going to induce a force to get away from each other. If the charges are positive and negative or opposite each other, those are going to be drawn towards each other. So two negatives would want to get away from each other, and opposites would attract. So the membrane potential has a negative charge on the inside, but as that negative potential on the inside is lessened with respect to the outside, there's a change in the force felt by the charges in the gate, and the gate swings open. And similarly for the potassium channel. Let's turn to our second channel type, and that's the ligand gated channel. In the first position of the channel, where here's the lipid bilayer again with the hydrophilic heads, and the hydrophobic tails, the cytosol side, the extracellular side. We've got more sodium on the extracellular side, just as before. But instead of a voltage activated gate, we've got a ligand binding site. And when a ligand binds to that site, the conformation of the protein changes such that sodium is able to pass through it and get from the extracellular side of the membrane to the intercellular side. Where do we find these ligand-gated channels? Well, there's multiple types of ligand-gated channels, but one example is at a junction, a neuromuscular junction or a synaptic junction. In this drawing here, it's a neuromuscular junction, and this is the terminal side of a first neuron, and then there's a space, and acetylcholine gathered up into vesicles is released into the space. That acetylcholine is going to bind to, for example, a nicotinic receptor, and that channel is going to open. Ions are going to flow into the cell on this side, raising membrane potential. All right, let's turn to our last type of channel, and that's a leak channel, which functions a lot like its name implies. There's no activation gate, be it voltage or ligand. The channel's always open, which doesn't mean it doesn't have specificity. There's still some filtering so that not any and all substances can pass through it. Some channels are specific to a particular ion, and of course the ions are only able to pass down their concentration gradient. Where might these channels be located? Well, it depends on the channel. For example, Hahn found two different sodium leak channels, and one type was localized to the entire somatodendritic compartment, while the second sodium leak channel was localized to the proximal dendrite ends. Let's see where that is. Now, turning to our picture of a neuron, we'll remind ourselves quickly that the leak channels, one sodium leak channel was present in the proximal, or these thicker branches of the dendrites, and the other sodium leak channel was present in all of the cell body and out to the distal ends of the dendrite. We also looked at voltage-gated sodium and potassium channels, which were present at the nodes of Ranvier along the axon, and we looked at a ligand-gated channel that would be at the adjacent end or across the junction of an axon terminal. Let's turn our attention to structure. Okay, now let's turn to the structure of our channel protein. And the first level of structure that we want to look at is that sequence of amino acids that forms our protein. And it's RNA that's going to dictate the sequence of amino acids, which ones and in what order. So if I started from the 5' end of RNA and moved toward the 3' end, where the outer perimeter is the 3' end, and I started with adenine, and then the next nucleotide was an adenine, and yet the third nucleotide was an adenine, that would indicate a lysine amino acid. And each amino acid is indicated by a series of 3 nucleotides or a codon. And you might notice that more than one particular series can indicate a given amino acid because there's fewer amino acids than there are 3 combinations of 4 of our nucleotide choices. So our first level is our sequence of amino acids, which are going to bond along their backbone. We can model them as a string of beads, but of course, amino acids aren't really just a bead. They have residues, they have side chain residues that might be positive or negative or hydrophilic or hydrophobic. And that's going to tend to move or change the conformation of our string from a linear string to its secondary structure, which could be a beta sheet or a helix, for example. It's a little difficult to discern this as a helix, but it's actually three-dimensional and forms a helix. The three-dimensionality of this sheet is easier to visualize because of the little folded piece of paper behind our amino acid chain. And this amino acid here is drawn to this amino acid here, and that's what's helping this string of beads transform into a helical shape. There's actually near 18 different secondary configurations where adjacent or nearby amino acids are bonded at some level to each other to form these secondary structures. And then the secondary structures, which also include loops and turns, is going to combine into a subunit, giving our protein its tertiary structure. And then combined subunits will form our final protein structure, which in this example here is hemoglobin. Looks kind of globular, works great for hemoglobin. It's floating around in the plasma inside of blood vessels. Then the last level of structure that we want to look at is the expression. And we recognize that a mutation in DNA is going to give us a different series in RNA, which may or may not end up in a different sequence of amino acids. But we know that a difference in amino acids might then affect the secondary structure as well as affecting its primary structure. But what can also happen with expression is we can synthesize too many channels. So recalling the voltage dependent sodium channel in the neuron that provides the upstroke of the action potential, if there's an upregulation or an overexpression of those sodium channels, that neuron can be hyperexcited and the person can experience, for example, seizures. What was surprising is Zhang found a mutant, so a mutation in the gene that encodes the voltage-gated sodium channel in neurons that results in a deficiency in expression of voltage-gated sodium channels. And in 20 to 30% of the animals that had this mutant, they also experienced hyperexcitability or seizures, which is unexpected considering there were fewer sodium channels. But what they found was concurrently there was a down regulation of potassium channels. Remember, those channels were co-localized in the nodes of Ranvier, and a decrease in potassium channels meant less potassium leaving the cell, less potassium repolarizing the cell, a higher resting membrane potential, and that higher resting membrane potential gave us that hyperexcitability and seizures in this underexpressed mutant. All right, let's move on to, let's return to our drawing of the ligand-gated channel, in particular the nicotinic acetylcholine receptor channel is shown here and has 5 subunits. If we were to look at the end or the extracellular view of this channel, it would have 5 subunits. Each one is shown in a different color. It looks like a pentagon. Each unit forms a side of the pentagon. One subunit is removed in this view so that we can see when a ligand isn't bound, the sodium channel isn't able to get through, but upon binding of the ligand, there's a conformational change in the structure of the proteins such that sodium is able to go down its concentration gradient. The transmembrane portion of these subunits of this protein is a series of alpha helices, and you can see the helices here. There's also parallel or similarity across the extracellular portion of the channel. Right. Let's look at a representation of the pore, which is neither just a funnel or a cylinder. It's going to have an extracellular portion, which would most certainly be a wide diameter. Then there'll be a filtering portion that, for example, would allow only sodium through it. There'll be a conducting portion of the pore. In other words, its function is neither to filter nor to collect, to draw ions in. And in the very bottom, in this particular channel, there'll be the gate that, when activated, will allow sodium, for example, to flow down its concentration gradient into the cell. Okay, and finally, let's look at the structure of a voltage-gated channel. In particular, we'll look at the voltage-gated channel. If we took a channel and cut it radially and stretched it out, we would have something that we could represent with this figure here. So this line here represents the cytosol side of the membrane, and the line above it, horizontal line above it, represents the extracellular side of the membrane. The channel is formed of 4 domains, Roman numeral 1 through Roman numeral 4. If you recall, in the ligand-gated channel, we had 5 subunits. Here we have 4 domains. Within each domain is 6 transmembrane segments. In between the 5th and 6th segment in each domain is the pore-forming portion of the protein. In between domains, there's extensive loops in the cytosol, and one is less extensive with respect to amino acid sequence, but is quite important because it forms the inactivation gate between domain 3 and domain 4. There's also a voltage sensing segment, and that's the 4th segment in each of the 4 domains. Now, looking at one domain in particular, we can see that it looks like each 4th segment has 2 charges on it, but that's just symbolic. There's actually 4, 5, 6, and 8 charges on the 4th transmembrane segment in respective domains. And if you remember from Coulomb's law, the greater the charge, the greater that force is going to be. So a 4 charge, all other things being equal, a 4 charged transmembrane segment is going to feel a smaller force than an 8 charge transmembrane segment, all other things being equal, with the same change in voltage. Now, these transmembrane segments aren't cylinders. This is just a representation, and neither are they something that looks like a spring. They are alpha helices, but they would look something more like this, with a helical background, but then all these side residues. And it's actually, for example, arginine that provides that positive charge sensor on the 4th segment. If we were to look at this channel from its end, instead of looking like a pentagon, it would look like a diamond, a 4-sided figure. And lastly, we just want to revisit the solid rendering of the core. The voltage-gated channel may look a lot like what we looked at previously with reference to the ligand-gated channel. Only one thing I wanted to draw attention to was in a leak channel, there is no gated portion. Great. Thank you for your attention, and references are to follow.